Good morning, viewer. Welcome to our FTV. Viewers, researchers at the University of Mansour University Bachon have demonstrated for the first time in real time that the body's own defense cell use catecholamines, that is called the neurotransmitters such as dopamine and adrenaline, to communicate via the same chemical signals as nerve cells. And this discovery opens up a new understanding of how the immune system is regulated. The study has been published in Advanced Science. Viewers, neutrophils are the most common type of white blood cell in humans and form the first line of defense against infection. The research team led by Professor Alois Erpenbeck from Munster and Professor Sebastian Kraus from Bochum had demonstrated that these immune cells process the same molecular machinery as nerve cells, and they take up catecholamines, store them in vesicles, and that is in small membrane bound vesicles, and releases them in a targeted manner in response to inflammatory stimuli such like neurons. We are surprised to see how similar neutrophils and neurons are in their ability to handle neurotransmitters. Erpenbeck wrote. Real-time observation was made possible by fluorescent carbon nanotube sensors, and these are tiny detectors that react sensitively to catecholamines and for the first time allowed their release from individual living cells to be tracked live under a microscope, where inflammatory signals, including serotonin and bacterial components, proved to be the triggers. Viewers, until now, we simply lacked the methods to visualize such processes directly in living immune cells cross-state. And what we have now observed using the nanosensors fundamentally changes our understanding of these cells. The released catecholamines have a direct effect on the immune response, and they curb an excessive defensive reaction by the neutrophils while simultaneously promoting blood clotting, a direct link between the immune and vascular system. Measurements of gene activity in healthy volunteers in whom inflammation was experimentally induced confirm that not only under in vitro conditions but also within the human body, neutrophils coordinate the adaptation of their catecholamine receptors and their synthesis and degradation program during an inflammatory response. Amazing, sir. Bow down. Benda's exclusive report.